Angiosperms. Unlike the gymnosperms where the ovules are naked, in the angiosperms or flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed by fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from tiny, almost microscopic wolfia to tall trees of eucalyptus, over 100 meters. They provide us with food, fodder, fuel, medicines and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons, figure 3.5. The dicotyledons are characterized by having two cotyledons in their seeds while the monocotyledons have only one. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. The anthers, following meiosis, produce pollen grains. The female sex organ in a flower is the pistil or the carpel. Pistil consists of an ovary enclosing one to many ovules. Within ovules are present highly reduced female gametophytes termed embryo sacs. The embryo sac formation is preceded by meiosis. Hence, each of the cells of an embryo sac is haploid. Each embryo sac has a three-celled egg apparatus, one egg cell and two synergids, three antipodal cells and two polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where two male gametes are discharged. One of their male gametes fuses with the egg cell to form a zygote, syngamy. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus, pen. Because of the involvement of two fusions, this event is termed as double fertilization, an event unique to angiosperms. Their zygote develops into an embryo, with one or two cotyledons, and the pen develops into endosperm which provides nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events the ovules develop into seeds and the ovaries develop into fruit. 3.6 Plant life cycles and alteration of generations. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generations between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups, as well as individuals representing them, differ in the following patterns. 1. Sporophytic generation is represented only by the one-cell zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant, photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represent this pattern. 2. On the other extreme, is the type wherein the diploid sporophyte is the dominant, photosynthetic, independent phase of the plant. The gametophytic phase is represented by the single to few-celled haploid gametophyte. This kind of life cycle is termed as diplontic. An alga, Fucus sp., represents this pattern. In addition, all seed-bearing plants, that is, gymnosperms and angiosperms, follow this pattern with some variations, wherein the gametophytic phase is few to multicelled. 3. Bryophytes and pteridophytes, interestingly, exhibit an intermediate condition, haplodiplontic. Both phases are multicellular. However, They differ in their dominant phases. A dominant, independent, photosynthetic, thalloid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant, independent, photosynthetic, vascular plant body. It alternates with multicellular, saprophytic, autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplantic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplentic, some of them such as Ectocarpus, Polysiphonia, kelps are haplodiplantic. Fucus, an alga, is diplantic.